record on this computer for this one. All right. So yeah, we'll record this and you can share it with your students, or you can edit out parts. What I'll eventually do is I'll convert it, put it into a YouTube channel, and then we'll link from the MCC website to the YouTube channel. So that way you can easily get it regardless of the format. You don't have to worry about logging in or anything like that. All right, well, let me share screen. Thanks for everybody for showing up. Abstracts, I was looking at the abstracts. Actually, I think I'm gonna stop sharing. I should have just shared the PowerPoint file. There it is. All right, so today's talk is not about the detailed kinetic analysis or the structural work with MDH. Later on, Ellis is going to do same kind of thing. He's going to have 2 or 3 of these sessions where he's going to talk about allostery and kinetics and things, and it's going to be broad. Later on, Lisa and Amy are going to put together one of these to talk about some of the techniques and the tips, hints, and tricks, if you will, of assaying MDH and some of those issues that folks are having trying to figure out what's going on in the class. This talk is really to talk about what's going on with human MDH, the differences between mitochondrial and cytosolic, understanding some of the regulation. I'm a cell physiology biochemist kind of person, so I take kind of a disease orientation to everything here, and you're going to get quite a bit of that. There's lots of things that are parallel here that are not disease oriented or not human or related that you could take and go to yeast. You can go to some of these other organisms. You can do plants. We have some really cool, interesting MDH clones available for everyone, but today is going to be really talking about the human MDH. So we're going to start with a little bit of background of what we have and just the basics. So cytosolic MDH, or MDH1, comes in a couple of different splice variants, all from the N terminus. Once upon a time, until just maybe about 3 or 4 years ago, there was very little information. There was no information that there were different splice variants of this. And now if you look at some of the PubMed and other websites that have all these databases, you can see there's actually messenger RNA for about 4 or 5 different splice variants of MDH1 or cytosolic MDH expressed throughout the human body. 2 of them are definitely active. Several of them are definitely truncations. And what does that mean? Are these things not active in terms of their enzymatic activity? Are they working as some sort of an anchoring protein or something like that? That's definitely a lot of cases for other proteins. And there's just very little information out there. So right now, I'm only focusing on 2 of the 3 most common splice variants: isoform 3, isoform 2. Isoform 2 is now considered, or I'm sorry, isoform 1. Isoform 1 is considered the canonical. This is the splice variant of MDH that's expressed in the majority of the tissues, not all the tissues. And there's quite a few tissues where MDH3, the longer term, we got about 16, 17 additional amino acids on the amino terminus of this express. This is expressed in neural tissues and a bunch of other tissues. In fact, we kind of found out about this because it was easily 9, 10 years ago. I took a cDNA library from human brain and then cloned out MDH, and of course turned into MDH, what we just called cytosolic MDH. It sequenced out, everything was great, it was active, it did the right things. But then later on, we found there were different splice variants, and it was the shorter splice variant that was expressed in more tissues than the longer MDH variant 3. So that was the one we've all started with. A number of you have used it before. Kate, you have used, I think, some cytosolic MDH, and that's where it started from. So then when we realized this, we used a student cure. Jess Bell helped out with some of her cures, and we did a couple of things. We added a TEV site between the end of MDH, the carboxy terminus end, and added a TEV site, then the histag. And then we also use that to turn around and make a shorter concomitant or canonical version, or MDH1. So we now have MDH3 and MDH1 sequence verified, active, well expressed, with each with, of course, a his tag and the TEV site after that. The TEV site's important, especially if you're going to try to do pull downs and the other proteins you're using has a histidine tag. You're going to want to remove histidine tag on one or more of the proteins. So prior to this fall, if you had one of the cytosolic MDHs, it was really clearly put in there that was MDH minus TEV. Now it's MDH plus TEV. So make sure you pay attention whether you have the older version or the newer version and splice variant 3 versus 1. I'm happy to send any of the above or all of the above to whoever needs them. So what do we have? MDH 1 and 2 are both homodimers. For the most part, our understanding, although it's kind of in the literature, there's some discussion or differences on how this actually happens. But the idea is that of the homodimer, one of the monomers acts as the active group and the other one is an allosteric regulator. Even though both active sites are fully functional, the second monomer acts as an allosteric regulator, which is thought to bind, citrate or other metabolites and change the conformation of both of them. These are available in a PET28 vector, which means if you're going to express them in bacteria, you have to make sure you have a DE3, BL21, or similar clone or a bacteria. They all, as I said, have TEV. The monomer size for the V1 and V3 will be 36.5 and 38.5. They express very, very well with an overnight 0.5 millimolar IPTG. 1 molar IPTG, a few hours at 37 degrees, you're going to get a lot of proteins. They're pretty stable. They're stable for several weeks at 4 degrees, long-term storage at 10 degrees or 10% glycerol with some salt at pH 8. You can freeze, but not freeze thaw multiple times, or you cannot freeze thaw multiple times. You can certainly freeze aliquots, take them out, and they retain over 90% of their activity if you have 10, even 20% glycerol in it. One caveat to all the MDHs, watermelon seems to be the one that's a little bit not in this case, but if you get much above 1 mg per mil within a day or two, and if you're really above 1 mg per mil overnight, you're going to have a snow globe. The proteins will aggregate and they'll fall out of solution, and they'll end up about 0.95 mg per mil when you're done. So that kind of tells you where that solubility limit is. If that happens to be you and you're working with these, there's all sorts of problems or questions on what remains in solution. But in a pinch, people have just spun down the precipitate and used the soluble remaining part with lots and lots of enzyme activity. We don't know what it looks like, but that's what it is. In fact, it expresses so well that in a liter culture, you can get 50 to 100 milligrams of the protein. I like to share websites that show recombinant protein purification, how much it costs per milligram of protein or microgram of protein, and then show them how much we just made. They think we've made the lottery when they see how much money we actually made in that 100 milligrams of protein. Unfortunately, there's not a market. We also have the human MDH cytosolic version cut into a PC-MVS vector, and on the C terminus of that MDH, we have flag and MIC dual tags, so you can do pull downs and other things if you want. For transient transfection, it has a marker, so you can actually make stable transfections from these transient transfections. It was tested once for activity or for expression, and they didn't get a good expression. It was done once with students in class, so I'm not really ready to say that it doesn't work because it came from the cDNA library from human, so we didn't need to change the sequence or anything. The aggregated published data I show here, I have a set of reviews on human MDH that talks about the kinetics, the structure, interactions, regulation, post-translational modifications. And these numbers are just an aggregate average of a number of different publications. So you can kind of get a feel for things. If you want those documents, let me know. They're fairly clean drafts, but they do need to be worked on a little bit before they're fully publicly available. So just let me know and I can hand that to you. So just recently, ACS published the first structure of human cytosolic MDH. You might think that's kind of weird, but for whatever reason, we've had pig and other cytosolic MDH structures, but no human. So just this year, somebody at the University of Minnesota expressed recombinant bacterially produced human MDH and shared the structure with them. And they shared their kinetics, which is not a typical way to show the kinetics where you have the concentration of the protein on the bottom and the velocity in millimolar of NADH consumed per second. So I think there's got to be some conversion to figure out how that matches our traditional. The Vmax should be okay. I'm not so sure about the KM, the way we typically use it, but you can at least find that here. What's interesting is, though, he does overlay the crystal structure this this researcher got with the mitochondrial human structure, which we do have, and they found there was a couple of different differences in the active site where they are binding some of the substrates. And in the paper, they talk about this might be an interesting place to do some drug design to hit mitochondrial versus cytosol or vice versa. And this could be a good place to do an MDH cure, to do some mutation or work with a synthetic chemist or something like that. That'll be one of the themes that I do today, is to talk about different spots where you can use papers or data to help your students start generating a hypothesis in their cures. I'm going to be focusing not so much on the pedagogy and how we're doing the cures, but maybe how we can use this information and you can start your own cure. So if you align the cytosolic and mitochondrial MDH, after you've added in, here's the cytosolic on the bottom, here's the mitochondrial. Mitochondrial MDH has that leader tag, which is not going to be found in the cytosolic MDH, because this is the targeting tag that brings MDH to the mitochondria where it's cleaved off. But once you align after that, you can see there's some areas with high domain, very much expected. You can see this structurally as well as in the primary amino acid sequence here. But there are some areas where there's some big differences in gaps, additional amino acids, that I think might be interesting to look at how MDH1 behaves differently than MDH2. And one of the first cures I did with MDH in the last couple of years was have students delete the mitochondrial sequence and insert the cytosolic sequence, and then start looking at changes in activation and inhibition and specific activities. And the idea was maybe we could find where cytosolic, mitochondrial MDH is the key sites for contacting citrate synthase. But there's a lot of good, simple, straightforward sequence analysis you can do to drive some hypothesis using something like this. And I can share all this information that you see here with you. Just reach out to me and ask. Of course, one of the reasons using citrate or malate dehydrogenase is because students will eventually see MDH in the Krebs cycle, and where they see, and I intentionally use this image from the web because they show this, of course, as a one-way arrow. In terms of the Krebs cycle, it certainly is, but not really, because if you show the thermodynamics of MDH going in this forward direction for the Krebs cycle, going from malate to oxaloacetate at standard state, it's not a very favorable reaction. It's plus 22 kilojoules per mole. So this thing is not going forward at steady state. So this is a point where you can bring students in and talk to them about coupling both in thermodynamics, because if you look at the thermodynamics of the rest of the cycle, clearly the next enzyme in place here that would use oxaloacetate from MDH is citrate synthase, and it's got a negative 31.5 kilodalton per joule delta G at standard state. So you talk about that. You can talk about how coupling these things with a physiological or non-standard state delta G could drive this reaction to go forward, but still separate from everything. The reverse direction is the more favorable one. So when you're thinking of cures, this summer when we had our in-person virtual cure with some of the folks in the new cohort, we were discussing the pH which should be used. And so I found a paper that compares heart human MDH from mitochondria and cytoplasmic. And what's interesting is they basically measured the rates of the reaction for MDH1 and MDH2, MDH1 seen in the light gray, MDH2 in the dark band here, and what they call mitochondrial conditions. So these are the concentrations that they determined in the cardiac tissue, NADH, NAD, everything was at, and at different pHs. And as you increase the pH, the rate not only increases, but we see an increase in the rate for MDH2, the mitochondrial MDH over MDH1. Sorry for this mistype here. And then the opposite is true for cytosol, again, using what they determined in heart cells to be the physiological concentration, that MDH1 is more active than MDH2 at these ratios. And as you go to a higher concentration or higher pH, the activity starts dropping. So we get a different effect there at the different concentrations. So just simply doing a KMV max at pH 8 is clearly maybe not, reflecting what we see physiologically, and will change the results. On the top of this, it's really actually hard to get not the pH range, but the NADH and NAD concentrations on things, because how you measure it, the tissue, the condition of the cells and the tissue when you extract the stuff. But the pH ranges in the cytosol 7, 7, 6, and that's kind of a big range. It's usually around 7.2 to 7.4. It can go all the way up to 7.8. So a pH 8 is really up at the high end of what you would find in the cytoplasm. And depending upon the acid state, if it's a tumor, it's being stressed, can get all the way down to 6.4 for short periods of time, not very long periods of time. One of the things I do with my other research is study intracellular pH. And if you get the cells down to 6.4 for very long at all, they're not going to be happy. Mitochondrial pH is a little bit more basic, 7.1 to 7.8. Maybe squeaking up to pH 8. So as you're thinking about doing a cure and thinking about doing the pH, thinking about the physiological relevance of this is important. Here I've got shared just from a couple of different sources, the relative concentrations, cytosolic, cytosolic, NADH, NAD, oxaloacetate, and malate. Our standard conditions for doing an enzyme assay is 100 micromolar NADH, 200 micromolar OAA, which, very different than the physiological concentrations. So what are some of the regulators that we know about in both mammalian MDH and others? And this is really important because, of course, this, as I said, is a dimer, and binding in one site will interact and alter the site of the second. Think of hemoglobin, think of ATCase, things like this. Well, citrate, if you look at the human literature, it's kind of controversial. It depends on the pH. It's inhibitory, especially as you get higher pHs, all the way at H. Well, we just showed you that we're not often at H, we're usually a little bit lower, so citrate might not be as inhibitory. It has been shown in different conditions to activate MDH going in the malate to oxaloacetate with a high enough citrate. So you get both inhibitory and activation depending upon the source. So there's a lot of interesting data that's out there, and there hasn't been a lot of, I think, thorough testing of how this is happening. There's studies that show the glutamate and alpha-ketoglutarate inhibit, malonate inhibits MDH activity. Higher concentrations of oxaloacetate, while looking at the kinetics going from OAA to malate, will start to inhibit the mitochondrial MDH. You see a pattern that goes up and then it starts coming down. We see whether it's a feedback inhibition or it's a second site that's now binding and causing a shift in the structure. Don't see that so much on the cytosolic versus the mitochondrial. Again, some interesting things to look at why that might be. Malate and NADH can alter the rate going in the opposite direction. So if you were looking at OAA to malate and add a little bit of malate, you can actually see an inhibition in the low end of the initial rates, looking at some interesting allosteric work there. Fairly recently, a group in Nebraska published some interactions between malate dehydrogenase and citrate synthase. On there, they showed that ATP, lower pHs, alpha-ketoglutarate, and succinate all increase the affinity of MDH for citrate synthase. Some of that intuitively makes sense, and some of it counteracts or contradicts the inhibition or activation that we see enzymatically. So trying to put these two things together, there's some really interesting areas of science in there. Meanwhile, coenzyme A citrate and NADH seem to decrease the affinity of the two proteins. So whether we're talking about allosteric regulators or the protein interaction drivers, even just the wild type, there's questions are out there. So why do we care? Again, because I tend to be, I got my biochemistry degree from, a medical school like some of us. I tend to be more biochemically, more clinically oriented. Again, don't have to be. There's lots of good reasons to study this for just the basic science. I've got nothing to do with disease, but I tend to kind of look at this. It helps motivate the students to give them a why do I care oidic moment. So MDH expression changes in cancer cells. We see some cancer tissues have mutations in the MDH gene, and we also see protein interactions are altered in hypoxic tumors. So HIF1 alpha and some maybe the pH of the cell can drive some of the protein interactions. And both in Alzheimer's and Lou Gehrig's disease or ALS have been published to show different kinetic results in the tissue from these samples. In a cancer journal a couple of years ago, they showed that when they did knockouts of either of MDH1 in two different cell lines with plenty of LDH and actin, that the cells without MDH1 had a decreased proliferation rate. They were doing these in Jurkat cells. They also use less glucose. It's not a shocker. When you show that in cancer cells versus normal cells in MDH1, you get more mRNA. So this came from a database that your students can actually study and then look at different cancers, different genes, including MDH. And they picked a couple of lung MDH diseases, cancer patients, and showed a modest increase in messenger RNA transcripts. They also looked at MDH1 and 2 in wild type cells, WI38, MRCs, and IMR90s are wild type cells, and they saw in a good number of cells, H522s, A549s, these are all cancer cell lines here, through Western blot, an increase. We also see an increase in MDH2 that sort of modestly goes along with the increase in transcript we see here. The survival of mutant MDH is correlated with patient death in a couple of studies. But what's interesting in this cancer journal cancer paper is when they literally homogenize the tissues and then measure gross enzyme activity, normal versus cancer cells, they get an 8-9-fold increase in overall activity. Well, that doesn't jive to the Western blot nor the messenger RNA data. So there's got to be something else besides just making more protein that's pushing some of this mutagen or the cancer cell behavior. I propose maybe post-translational modification. So if we look for parallel studies, another place to have students look at and maybe read some papers as they think about their work or design their hypotheses is to look at two enzymes, pyruvate kinase, the M2 variant, and lactase hydrogenase. One, they both involve MDH, but more importantly, you can see how changes in post-translational modification change the behavior of the enzyme in a pathway that involves MDH. So if we just start with what we call the Warburg effect, protein kinase M2 becomes a predominant protein kinase, I call it protein kinase, pyruvate kinase in glycolysis in a number of different tissues, and it supports the Warburg effect. The idea is when pyruvate kinase M2 is a tetramer, it's active. In various cancer states, MYC and other protein kinases will phosphorylate this, turning it into a dimer or a monomer with a diminished function. That function is important because that stops the glycolysis pathway at that point and pushes both glucose and reduction potential over to NADPH and ribose and pyruvate to support cell growth, making new nucleotides and other building blocks for these actively growing cells. What's interesting is when that happens, we look at this pathway down here. In glucose, if we stop because of pyruvate kinase at bisphosphoglycerate, we run out of reducing potential, right? And so at this case, we're not getting as much TCA cycle. So MDH needs to be involved to replace those reducing potentials so the glycolysis as it is can continue. That means citrate from the mitochondria are transported out using ATP citrate lyase to make oxaloacetate. Malate is then converted to pyruvate through malic enzyme. That pyruvate can then be cycled in. So now we have a different cycle, which you might not have seen very much of before, that involves MDH because of pyruvate kinase. But the other takeaway for this is that you phosphorylate pyruvate kinase, you change glycolysis in a way that helps drive cancer formation. Does that happen with MDH? Interesting, wouldn't it be? Plus, this also suggests that MDH in the cytosol has an OAA to malate direction and might interact with malic enzyme and ATP citrate lyase. In gluconeogenesis, in glucose cases where we start finding in cancer cells, glutamine is converted to glutamate and then metabolized to alpha-ketoglutarate. It has two functions or sources. Alpha-ketoglutarate will be converted to citrate, which will then be used in this pathway I just discussed. Also, alpha-ketoglutarate will then go through fumarase to malate dehydrogenase. So the idea that MDH is going to then interact with citrate synthase to make oxaloacetate actually doesn't work in this case. So any interaction between MDH and CS, we would actually want to see in a cell break that interaction so that way the malate doesn't continue on through the Krebs cycle, but it could shift out into being used for metabolic building blocks in cancer. So notice in here, we have different functions of MDH, silent or less quiet in mitochondrial and a very active cytosolic. We look at LDH, we find that LDHA is another variant of lactate dehydrogenase, and it's phosphorylated in a number of cancer cell lines. And this phosphorylation, interestingly, not only changes its activity in support of glycolysis in cancer cells, but once LDH is phosphorylated, LDH will go to the nucleus and start driving the gene expression for cancer-specific genes. I didn't say this before, but when pyruvate kinase is phosphorylated and it becomes an inactive dimer or monomer, it also translocates to the nucleus. Did I mention there's a truncated MDH2 that doesn't have an active site? I wonder what it might be doing. Does MDH2, again, when it's phosphorylated, do some of this as well? Good question. So MDH is involved, of course, in a number of different classic and well-known pathways and creates a nice cure to look at things. This is complicated here. I want to break down to kind of show you why maybe MDH needs more fine-tuning or regulation than just whatever substrate happens to be present. So if we look at the proteins that share either the substrate malate or oxaloacetate in the mitochondria, we have fumarase, we have citrate synthase, glucose oxidase transferase, and pyruvate carboxylase also can interact with or can share either oxaloacetate or malate. And if you look at some of the protein interaction databases, each one of these is predicted or has some evidence of interaction, usually in massive pull-down mass spec type studies. The same thing can be seen if we investigate the enzymes that share either product or reactant, oxaloacetate or malate with the cytosolic MDH. We got malic enzyme, another glucose oxidase transferase, ATP citrate lyase, and phospholinopyruvate kinase. But how can these three enzymes compete for binding oxaloacetate at one time? David Heck at Community College here in San Diego with his freshman class did mole modeling and found most of these guys bind near or at the same site or predicted to bind in his complexing path software, binding of what might be near the same site. So is, do they all bind at the same site? Is there something that would affect one binding affinity over another? Hard to say, hasn't been done. To really bring this home, if we look at what MDH1 is doing in each one of these kind of pathways, in gluconeogenesis, in the cytosol, MDH is going oxaloacetate, malate to oxaloacetate, and potentially interacting with PepCK during the malate shuttle. Same direction, but would preferably interact with glucose oxidase transferase to support that pathway. In amino acid metabolism and with the TCA intermediates, we have the reverse, MDH interacting with GOT1, but in reverse to then make pyruvate through malic enzyme. And in adipogenesis, ATP citrate lyase, MDH1, and malic enzyme to produce pyruvate. Mitochondrial, in Krebs cycle, we all know malate to oxaloacetate, but in gluconeogenesis, pyruvate coming in converted to oxaloacetate from pyruvate carboxylase needs not interact with citrate synthase, but be released as MDH. And you can see over and over again, this is an interesting pathway that happens in a number of cells where we're swapping NADH for NADPH. And so we've got these three enzymes in a cycle getting NADPH when we need to do some biosynthesis. And of course, glutamine synthesis. So again, we have MDH needing to interact with different proteins or break interactions to allow different metabolitic outcomes to happen. What's driving that? Well, two things could be driving it. Metabolic intermediates that act as allosteric structural changes, and of course, post-translational modification. Another interesting aspect of MDH, if you're interested in disease, is what MDH is doing as an oncometabolite. So hydroxyglutarate, both D and L, are oncometabolites. We see these produced in a number of different cancers. It's actually becoming a marker for a number of different cancers. They're involved in histone and DNA methylation and control of a number of things. So the main process is that isocitrates getting converted alpha ketoglutarate, and then through either LDHA or MDH1. LDHA is the version of lactate dehydrogenase that increases in cancer, and we know that MDH1, cytosolic MDH, will make 2-hydroxyglutarate. IDH will, a mutant version of it, and another enzyme can make D-hydroxyglutarate. Well, then hydroxyglutarate will then interact with other proteins. We can also make it through the mitochondria. So we can go D and L through MDH 1 and 2, but very little work's been done on that other than it's happening. And is there a time where this is a minor pathway that this might become a major pathway? So what my students have been doing over the last couple of years, both in and out of CURES, is to look at the databases, the mass spec databases. We started with, well, let's do the prediction software. And then we found there was enough information in the experimentally defined path databases where we just ignored the predictive enzymes that would phosphorylate either MDH1 or MDH2. And these are big mass spec databases where they do tissues, mammalian tissues, human tissues, and others, cell cultures in different conditions. So there's no real understanding of what percent of MDH is actually being phosphorylated, what are the conditions that they're phosphorylated or not. So my students over the last couple of years, including this summer, looked at these databases, and especially if they were in 2 or more different sites showing up to be phosphorylated, we counted them. So we were being pretty tight in our screen. Plus we used the more stringent screenings when we were picking which phosphorylations. And you can see here, normally we would just lump serine and threonine together, but it's interesting that overall we got 22 experimentally defined cytosolic MDH sites of phosphorylation and 38 for MDH2. Phosphoserine has 6 or so in common. There's no common sites for threonine or phosphotyrosine. One of my students is now being able to try to do some predictions at the consensus sequences and using some other SAT databases to try to predict some of these kinases that might be working at this. But MDH is also post-translationally modified by acetylation. 3 in the cytosol and 15 sites in the mitochondria. For those who aren't really aware, if we take a look at lysine, when we acetylate it, of course, we're changing the charge, we're changing the nature of the non-covalent interactions that happen when you get acetylated. I'm sure most people have heard about acetylation of histones. This is a reversible post-translational modification that has been experimentally defined in MDH. And in terms of doing a cure, you can see that there are some phospholimics for phosphorylation. So in aspartic acid or glutamate, somewhat phospholimics of phosphorylase serine and threonine. There's not a good phosphorylation mimic for tyrosine. You can do a phospholimic for acetylate, or a mimic for acetylation with a glutamine or an arginine, and then methylation. There's a couple of papers that show MDH2 is being methylated. Well, again, these are helpful but not definitive site-directed mutants that you can ask a lot of different questions of where it's being methylated. So there's a wide open field here of these mutations, or I'm sorry, these post-translational modifications, and only a couple of these acetylations and a couple of the methylations are in the literature at all. None of these phosphorylation sites that are out there in these big mass spec databases have any papers on them. What's it doing to this expression? What's it doing to the half-life? What's it doing to the kinetics, the interactions with allosteric regulators like citrate? Is it driving or defeating protein interactions? There's literally no data out there, except for to see that it's increased in its activity in certain cancers. So there's a lot of juicy space to do work on. Last spring and this summer, some of my students were purifying and looking at MDH2 and doing some of this phospholimic direction where we looked at 4 sites. We looked at the melting curves and we found the phosphorylation really changed the stability of the protein. If we look at MDH2, there's a major and a minor TM. Looking at just the major TM, you can see that when we change S45, T85, threonine, serine 222 or threonine 224, we get a couple with some significant changes in the melting point. What's interesting is the overall specific activity changes quite a bit. We see that S45 drops the activity by about half. T85D doubles or more the activity, while S22 drops like a rock this activity. And at the time, I kind of argued with my students, why are we looking at 222 and 224? They're right next to each other. They're going to do the exact same thing. This is boring, but they really wanted to, so I caved. And this is really interesting. The CURES students got this, and then my summer research students started getting this. And then what's interesting is another person doing human MDH, but with the cytosolic, did this similar 2 sites, and she saw the same thing, that one knocked out and one increased the activity significantly. So there is some really interesting data out there on this. Again, if you're interested in what's the difference between cytosolic and mitochondrial, we have this laid out. We have the phosphorylation sites bolded. We have, at this time, we only had 4 sites that we were making mimics. I now have, I think it's 28 different serine or threonine to aspartic acids for mitochondrial, and some of my students just designed 6 for the cytosolic MDH. So once we've got it made and validated for sequence, if people want to use them with their cures, we're ready to share. Talked about methylation and acetylation. We'll kind of move on from there. So what do these phosphorylations do to the activity of the enzyme? You saw with that one study a lot. How those protein phosphorylation sites regulate the allosteric, because with pyruvate kinase, it normally is driven to the active tetramer when fructose 1,6-bisphosphate's around. When this protein kinase or pyruvate kinase is phosphorylated, it goes to the dimer in active state And it doesn't matter how much fructose one or bisphosphate's around, that activation is lost. Do some of these things in parallel happen with all these phosphorylations we're looking at? Maybe some of them are not very physiologically relevant, but we don't know until we test it. So the other part is, what about protein interactions? We want to know where the proteins interact. Using this to drive a cure is also really interesting because it makes students think about structure and function and why does a protein interact with another protein? They don't want to do anything. It's non-covalent, it's kinetic affinities, KDs, KAs, this kind of thing. For a long, long time, we've known that MDH and citrate synthase are coupled. That means that there is some interaction that shuttles malate to oxaloacetate in the active site of MDH, and that oxaloacetate through kind of a ditch, if you will, not a channel or a tunneling, but a positive charged surface of the two proteins keeps the negative charge oxaloacetate bouncing along to get to the active site of citrate. And the idea is there's lots of studies showing this is happening, but we still don't know where they're happening. The old school studies would take and do size exclusion chromatography. And one of the early studies, which I kind of like here, they would run citrate synthase through the protein and then add malate dehydrogenase and look at the shift alone and with citrate synthase. And of course, when citrate synthase and MDH were together, came out much later in elution fractions for the size exclusion chromatography. And they didn't see this interaction when they did the same thing with cytosolic MDH. But later on, somebody looked at a different way of showing the interactions using fluorescence anisotropy. And aldolase, there's not a lot of good controls for these things. You use BSA, glom onto everything, mycelium fats. They used aldolase and citrate synthase. They used mitochondrial MDH, citrate synthase, and saw, as you titrate in the second end protein, you saw that fluorescence anisotropy change. But look at this, the cytosolic had some interaction. The authors literally ignored that line altogether. But to me, that means there's a little bit of affinity of cytosolic MDH with citrate synthase. And in one of my students' earlier studies, they saw that with some of the melt curves, a minor shift, but not as large as a melting curve you see the mitochondrial MDH. Another way you can measure this in a CURES, and you got to be really careful, is to have a mixture of MDH and citrate synthase, and then a competing enzyme like maybe GOT, aspartic transaminase. And if oxaloacetate is fully transferred from one active site to the other, there's very little leakage of MDH or oxaloacetate to then interact with GOT. Got to be a little careful with this because there's indications that GOT in the mitochondria will interact with both CS and MDH. So you have to use maybe the cytosolic version. And that was earlier than this experiment came out with. This experiment also shows the importance of crowding agents in some of these activities, PEG, FICOL, things like this. So a few years ago, a group or two groups cross-linked proteins and then modeled them. They took citrate synthase and MDH, cross-linked the lysines, cleaned it up, threw it through mass spec and said, who's talking to who? What lysine is talking to what lysine in between MDH and CS? What was interesting and confusing still to me, at least at this point, if they took commercially purified porcine MDH from Sigma, they got one set of interactions or crosslinks. If they used bacterial crosslinks, not only to get proteins to do the crosslink, not only do you get less, but they're not 100% in union with the other. And then if they cross-link gross tissue from beef tissue and then did the mass spec, they found a lot less crosslinks, which is not that surprising. But the fact that every one of these crosslinks doesn't exist in the other is confusing. Is there something happening that we see in this preparation that doesn't happen here? There are a lot in common, but not all of them. Well, when you dock these, and this again is docking, it's interesting. It helps drive maybe where that interface might be. In that publication, they used CLUS Pro and they looked at recombinant protein docking, and they used those those cross-links, they came up with this as their top dock because they restricted the docking to the sites where they knew that lysines would cross-link. They did the same thing with the bovine and the same thing with the sigma. And if you look at the delta G of association, they're, one, you can clearly see they've got different faces with each other, and two, their predicted delta G using HawkDoc, GGMBSA, very different. We use the handful of common cross-links and clean them up, and then use both ZDoc and ClusPro and came up with this orientation. Again, this is just docking. This helps us try to figure out what's in the interface. When you use human MDH and CS with HawkDoc or MO without any limitations, you get, again, different orientations. So take it for what it is, but eventually you got to try to start guessing where that interface is and use this to make your priorities. So one of the things we need to do is get better pull down assays. So if somebody is good at pull down assays with weaker affinity proteins, we need to come up with a good, robust plan for doing this in a cure. I did this in my cure. We saw the interactions, but they weren't near as strong. We need to go back and really flesh this out. I keep saying this. We need a couple of people that are willing to work together and do this. So what are the problems out there? Oh, good Lord, there's too many to count. We got cytosolic MDH interactions. We have Pepsi K, GOT. We have malic enzyme that are available. We could, with under a few $100s, make ACL or pyruvate carboxylase as well. I have 3 different mutants of TEV that should be a better version of TEV that could even cleave TEV off of the column, which is a problem with turbo TEV or wild type TEV. We've already expressed these. We found good expression levels. We see they cleave, but we haven't played with that combination yet. And the TEV, recombinant TEV, would be nice to have to get in bulk for people to share and use in your cures rather than making them. So there's a nice little cure here, or even a research project for somebody. We see things are acetylated, methylated, phosphorylated across all sorts of different species. What's going on between different species? It doesn't have to be limited to human cancer or something like that. There's a lot of cool in vivo action. Use the PCMBS to stick MDH in there, do a pull down. Under what conditions is it being phosphorylated? How does that activity directly align there? Do a serine to alanine, do a tyrosine to a phenylalanine, transfect it, pull it out using the MIC flag tag, and then measure the activity, throw it through mass spec. It's that easy, right? No, that's not the case. There's a lot of interactions that are available that we can do. There's simple kinetics, the mobile loop of the binding pocket of cytosolic and MDH. We now know there's some differences that the author suggests would be really an interesting place to design a drug to. Well, designing a drug to would be interesting there. Pick MDH1 over 2, or play with those sites and see how important those differences really are. Nobody has yet, and I'm one of the students on this, Olivia and Shri, if you're out there, we need to do this. What is the effect of the longer and shorter amino-terminus splice form 1 and 3 on the kinetic analysis? We don't know. Nobody knows. It's never been published. And we just finished making that clone this summer or this fall. So it's there. There's a whole cure right there. Allosteric, binding, interaction structures, you name it. Phosphorylation, what kinase is doing what to who? This is a good Southern song here happening. What are the contexts of which this phosphorylation is doing it? Does the phosphorylation act on the allosteric regulators like citrate and things? Is there a pH responsive difference? What are the interacting partners, and so on and so forth? So before I go on any further, is Delphine here? Okay, Delphine thought she might be able to come. So Delphine also works, she's a new to MCC. She started in the cohort this year. So I'm going to show a couple of her slides that she's doing with this. So Delphine at the University of Vermont, she was there this Saturday. She brought some of her students and presented what they were doing. Another post-translational modification, there's a whole bunch of them besides phosphorylation, acetylation, and methylation is ADP ribosylation. Pyruvate kinase is also ADP ribosylated, and that changes its effect and actually can drive it to the nucleus. It's a reversible post-translational modification. PARP is the enzyme that does this. You can get macrodomains that interact with the ribosylation of the protein. You can actually get multiple ribosylations happening on a protein. And this looks like it's happening on MDH. She's looking at a number of different enzymes that would potentially ribosylate MDH1 and MDH2. There's some data that she has that indicates that the ADP ribosylation of MDH1 and 2 is actually taking place on what residues she's starting to look at. There's some inhibitors of PARP, so you could do this in cytosolic or in culture cells as well as doing the recombinant proteins. So she's really trying to understand in her cure how the ribosylation of MDH, what it does to structure and function, what's happening to MDH when it's being ribosylated. Could be very different. Could it change the structure of it? Does it change what MDH is interacting with, right? There's a lot of really juicy interacting things here. So what time is it? Oh, I almost went the entire hour. At this point, I want to open it. If people want to talk about what maybe they're doing, we can do that. There's a lot of collaborations. We collaborate, and that's probably one of the, I think, the nicest things about doing these kind of MDHs, cures, is I'm working with Amy Springer, and they're not doing any phosphorylation stuff. They're doing something very different. I've worked with Kate. I've worked with others, people. Ellis has been, Jess has been, Zing's. It's been some really cool interactions. Chris and I are working with our students right now and looking at how MDH and citrate synthase and maybe these phosphorylation sites impact the interaction using SACs. And we've got our initial look at it. I'm really excited because I'm not a physical biochemist, and I saw that and I saw magic, Chris. So at this point, any questions? Anybody interested? Does somebody want to maybe share what they're thinking about doing? Is anybody would like to use some of these reagents?